Assalamu alaikum class. We are going to discuss functions of liver today. Function of liver are as follows: metabolism of carbohydrates and lipids. Second is deamination and urea formation. Third is production of bile that takes part in digestion of fats. Fourth is detoxification. Fifth is formation of cholesterol. Sixth is thermoregulation, that is actually distribution of heat throughout the body. And seventh is storage of vitamins. Metabolism of carbohydrates and lipids as a first part. Liver takes part in the metabolism of carbohydrates and lipids. It regulates the amount of glucose into the blood, and an extra amount of glucose is converted into glycogen, which is a reserve food material. The glycogen also be used to provide energy at the time of need. When the stored glycogen is reduced in size, then amino acids are converted into glucose by liver. Liver also helps in the oxidation of lipids, or these are removed from the blood and changed them into fats, which are stored in the body. Liver also prepares unnecessary amino acids and plasma proteins, for example, prothrombin, fibrin, and globulin, etc. Deamination and urea formation. The excess of proteins and amino acids cannot be stored in the body. Proteins are changed into amino acids, and from amino acids, ammonia is removed. The process called deamination. After deamination, the remaining part of amino acid is used in carbohydrate metabolism to supply energy. Ammonia combines with carbon dioxide and converts into urea by a cycle called ornithine cycle. Important steps of ornithine cycle are: ammonia combines with carbon dioxide to form citrulline. Citrulline is converted into arginine by addition of some more ammonia and removal of water. Arginine is then converted to ornithine by removing urea and addition of water. The cycle is repeated again and again, and urea is formed again and again. The third role of liver is production of bile. Liver produces a secretion called bile. It is yellow-green alkaline substance. Bile is transferred from liver into duodenum. It neutralizes the acidic chyme, and bile is also antiseptic and acts as germ killer. It also combines with food, and it is also antiseptic. That means it kills bacteria. It takes part in the emulsification of fats. I will go a bit very fast because while recording a lecture for you on YouTube, I have twice deleted my lecture videos by mistake, and I'm just not very happy with this progress. So I'm just going to give you an overlook of this topic. Detoxification. D for deletion. Toxification. Toxic is for poisonous substances. Liver converts toxic substances into non-toxic substances. It changes the structure of medicine and poisonous substances. This process converts these materials into harmless substances. For example, a compound, hydrogen peroxide, is a harmful compound. The liver secretes an enzyme, catalase, which converts hydrogen peroxide into hydrogen and oxygen and makes them non-poisonous substances. Formation of cholesterol. Liver also forms cholesterol, which is necessary for the body. Its extra amount is always excreted along with water. The extra amount of bile causes stone in the gallbladder and causes jaundice. Thermoregulation. Liver helps in regulation of body temperature by continuous supply of blood and metabolic processes. Storage of enzymes. Liver also stores enzymes such as A, B, and D. It's actually storage of vitamins. Liver stores vitamins such as A, B, and D, especially vitamin E, vitamin B, and vitamin D. Please correct this mistake. And the next part is urinary system of man, and that is very much familiar to all of you. We know human has got kidneys as an excretory organ. They have got ureter on both of the sides. Then comes urinary bladder and urethra that expel this urine out of body. Here comes an artery that's called renal artery. The other is vein that's called renal vein because the suitability of this kidney. And these glands are called adrenal glands because they are added to renal. That's why they are named as adrenal glands. Excretory organs consist of kidneys, ureter, urinary bladder, and urethra. Structure of kidney is a pair of dark red bean shaped structures which are attached to the dorsal wall of abdominal cavity. They are covered ventrally by peritoneum. Peritoneum is a bag or layer that covers them. Like we know, pericardium is the protective covering or bag of heart. Meninges is bag of brain, so peritoneum is bag of kidney. Here we come. Usually embedded in certain amount of fats. The outer side is convex and inner side is concave. The inner concave side has a notch called hilus. From the hilus, a white tube arises known as ureter, which passes backward and opens into a thin-walled pear-shaped sac called urinary bladder. From urinary bladder, a tube-like structure is produced called urethra, that is the part in the excretion of urine that takes part in the excretion of urine from the body. At the end of urethra, there is an opening which controls. There is a sphincter present, which we call a valve that controls the excretion of urine from the bladder. Internal structure of kidney is internally the kidney consists of two parts, an outer cortex and inner medulla. The kidney contains a large number of very fine tubules called nephrons or uriniferous tubules. These nephrons are about 1 million and they are held together by connective tissues. In medulla, many cone-like structures are present called pyramids. The pyramids open into a wide funnel-shaped part called pelvis. From pelvis, a narrow tube ureter arises, which carries urine to the urinary bladder. It stores urine for a short period, then it passes through a narrow duct, urethra, from where it is discharged out of the body. This is the structure of kidney. Pelvis, medulla, this is nephron. The cup-shaped structure is an outer area, tubules are inside, and then they are excreting, collecting urine. Here, this point is hilus, this is ureterization, and this ureter is collecting all the urine collected from all medulla and pyramids, and it's taking it to bladder. This is another very fine structure, ultra refined structure of kidney, human kidney. Let's study about this. Structure of nephron. Each nephron consists of three parts: Malpighian body, loop of Henle, and collecting duct. This is Malpighian body, loop of Henle, and this is collecting duct. Malpighian body consists of two parts: Bowman's capsule or glomerulus. It's here. It's a nephron. This cup shape is Bowman's capsule. This cluster of arterioles is called glomerulus. This convoluted tubule is called loop of Henle, and this is collecting duct. Number 1, Bowman's capsule. The upper part of nephron is cup shaped, called Bowman's capsule. It has glomerulus in its cavity. Number 2, glomerulus is a cluster of branches which is present in the cavity of nephron. It is formed by a branch of renal artery called afferent arteriole. The one that is coming to tubule is afferent. The one that is leaving glomerulus is efferent. The blood is carried away from the glomerulus by a small vessel called efferent arteriole. The efferent vessel forms two types of capillaries, proximal and distal. Here you can see one is proximal, other is distal. Let's see. Another capillaries are present around the two limbs of loop of Henle called vasa recta. Below the Bowman's capsule, a small proximal convoluted tubule is present, which is attached to the loop of Henle. Malpighian body and proximal convoluted tubule are found in the cortex region of kidney. In Bowman's capsule, filtration of blood takes place from glomerulus. Now comes loop of Henle. The proximal convoluted tubule opens into a U-shaped structure. It passes down into the medulla, where it bends back and runs up again into the cortex. See, you can see it here. Come, go, and then come back. Okay, descending and ascending limbs. Descending, coming down, ascending, going up. It has two limbs. It checks the concentration of urine. It is the point where reabsorption takes place. The ascending loop of Henle opens into another convoluted tubule called distal convoluted tubule. This tubule opens into collecting duct here. Collecting duct, when the tubule reaches the cortex, it again becomes convoluted and then passes into a straight part called collecting tubule. Many collecting tubules join to form the discharging tubules, which finally open at the apex of pyramids. They lead into pelvis, which is the expanded another part of the ureter lying within the kidney. Okay, this was the structure. Now we can see that how regulatory function of kidney are being performed. The mechanism of excretion consists of following steps. Number one is ultrafiltration. The second is reabsorption. Third is tubular secretion, and fourth is countercurrent exchange. Ultrafiltration, it occurs in the Bowman's capsule. In the process, all the substances present in blood are separated from it, such as sugar, salts, nitrogenous compounds, and water. Filtrate is transferred from glomerulus into the Bowman's capsule, and pure blood remains in glomerulus. Reabsorption, when the filtrate passes through the nephron, reabsorption takes place in the following parts: in proximal convoluted tubule, in descending limb of loop of Henle, in ascending limb of loop of Henle, in distal tubule, and then here. First is in proximal convoluted tubule. Here from the diagram you can see that in this part reabsorption of glucose, amino acid, water and salt take place. Descending limb of loop of Henle, when the filtrate passes through the part of nephron, water and sodium are reabsorbed. Other substances are not absorbed in this part. Third is ascending limb of loop of Henle. In this part, sodium and chloride ions are reabsorbed, which are transferred to the interstitial fluid and then enter the blood. The reabsorption of sodium chloride ions occur by active transport. Here water is not absorbed. Distal convoluted tubule. From ascending limb, the filtrate comes into the distal convoluted tubule. It is slightly hypotonic due to high concentration of water. In this part, by the action of hormones, concentration of various salt is adjusted. By the adjustment of concentration, the filtrate now becomes urine. It is passed into the collecting duct. In the collecting duct, the concentration of water in the filtrate is also adjusted. Then comes tubular secretion. Tubular secretion is also an important process in excretion. In the proximal and distal tubules of nephron, some harmful and nitrogenous substances are transferred from blood into the filtrate by active transport. These harmful substances are ammonia, urea, uric acid, and creatinine. These are the substances which are not separated from the blood in Bowman's capsule. Hydrogen and ammonium ions are also secreted from interstitial fluid into distal convoluted tubule. It maintains acid-base balance in blood. Countercurrent exchange. The exchange of solutes and water in the medulla of kidney is called countercurrent exchange. There are two systems of countercurrent exchange. One is countercurrent multiplier, other is countercurrent of vasa recta. Countercurrent multiplier occurs in the loop of Henle and maintains concentration of ions in the medulla. When the filtrate passes through the descending limb of loop of Henle, water is reabsorbed. Due to the process, filtration becomes more concentrated. Filtrate becomes more concentrated, that is hypertonic. When this filtrate comes into ascending limb, the sodium and chloride ions are reabsorbed from the filtrate, so it becomes dilute, that is hypotonic. By this process, in the inner medulla of kidneys, high concentration of solutes, that is sodium and urea, is maintained. Due to hypertonic condition of filtration in descending limb, about 90% water is reabsorbed in the ascending limb. So concentrated urine is passed outside the body during excretion. Countercurrent of a system of vasa recta, the blood vessels which run parallel to the loop of Henle are called vasa recta. Through these blood vessels, only 10% of the blood of kidney passes very slowly. This blood supplies oxygen and nourishment to the cells of medulla and carries away the reabsorbed water from the filtrate. In this way, the system is regulated and maintained properly. Then next topic comes adaptation of kidney and types of nephrons. There are two types of nephron: cortical nephron and juxtamedullary nephrons. Cortical nephrons are like this. They are found in cortical region. They all of their three of the parts: Malpighian body, convoluted tubule, and proximal tubule. All the three parts are in cortex region. That's why they are called cortical nephron. The other is juxtamedullary nephron. Their capsule is here. The convoluted tubule is here, but their proximal tubule, loop of Henle, is within the medullary region. That's why they are named as juxtamedullary nephron. Let's read the text. In animals, the structure of kidney is according to their body and the environment in which they live. Due to the shortage of water, the body of animals have the system to retain water and prevent its over excretion. The concentration of urine depends upon the length of loop of Henle in the nephron. When the loop of Henle is long, more water can retain in the body and concentrated urine is passed out of body. When the loop of Henle is short, then dilute urine with more water is removed from the body. There are two types as we mentioned. The first is cortical nephron. These nephrons are short in size. The loop of Henle is reduced and present only in cortex region of kidney. The very small part is reached to the medulla. Juxtamedullary nephron have long loop of Henle. These are present in medulla of kidney. They reabsorb more water which is required in the body. It is according to the need and size of animal body. This is it for this lecture because it's exceeding the allowed duration. I am going to put a remaining topic of this chapter in the coming lecture, which inshallah be the last lecture of this chapter, and then we will be starting another chapter. As I have received some requests from students that they wanted to have revision of genetics topic first, so we will be doing genetics first, genetics and ecology. These are those chapters which we did in the end of our academic year, and their notes are not yet dispatched, so I will try my best that I've got the soft copies of those notes. I'll be forwarding notes along with the lectures. Inshallah, but to see you in the next lecture. Keep visiting Facebook page, WhatsApp group, and do respond us so we will get to know what there may be the measures we can take to improve our productivity. Thank you very much. Stay home and stay safe. Thank you, beta. See you. Jazakallah.